Hi, today we're going to talk about the Clostridioides difficile PCR-LAMP test. This video was made by Redondo Maya Geraldine, Hernandez Lopez Simon, Cortés Michel, and Ortiz Bautista Abril.
Clostridioides difficile is a gram-positive bacillus, strictly anaerobic, spore-forming, and toxic-producing that exists in the soil and gastrointestinal tract. In 1978, it was associated with antibiotic-associated diarrhea, toxic megacolon, sepsis, bowel obstruction, and abdominal perforation. The pathogenicity comes from the production of the enterotoxin A and the cytotoxin B that produces inflammatory reactions in the intestine. They can be detected with a cell culture assay or different enzyme immunoassays, as well as the rapid and specific loop-mediated isothermal amplification, or LAMP, is developed to diagnose infectious diseases. The LAMP technique, it uses 6 types of primers to detect 8 regions of DNA using a polymerized enzyme with field-shifting activity that detects these DNA sequences and can be done at a temperature of 63 to 65 degrees Celsius.
The strain Clostridioides difficile associated infections are toxigenic because of their ability to produce cytotoxins type TCDA and TCDB. This type of strains have pathogenicity locus, PA-log. Most of PA-log have 5 genes and one of them codifies for TCDA and another for TCDB. Both toxins get in the epithelium cells and producing the glycosylation of RHO proteins, inactivation of this protein. When it happens, it generates the desegregation of microfilaments of actin and redistribution of time junction proteins like occludin, making in this way an alteration of epithelial barrier and blocking the phagocytosis of vegetative cells of Clostridioides difficile. After this, the mechanism of TCDA consists in adherence on the apical surface of colonic epithelium, especially in the glycoprotein GP96, and translocate the protein to the cytoplasmic cell, producing the liberation of pro-inflammatory cytokines like leukotriene, Vg, and alpha necrosis tumoral factor. Then, it activates the liberation of IL-1 and IL-6 from monocytes. It makes the migration of neutrophils. On the other hand, for TCDB, it crosses to the lateral basal cells, and then it adheres on a receptor. This receptor is still not recognized. Both toxins activate the enteric nerves, making an elevated production of neuropeptides like P substance and neutrosin. They provoke the secretion of a lot of chloride on the epithelial cells of the intestine. Using the PCR test in a sample of the stool of the patient, it is possible to identify genes from Clostridioides difficile and now what type it is, the producer of TCDA or TCDB. The PA-log contains genes for 4 additional proteins, TCDR, E, L, and C, which are thought to play a role in the regulation of toxin expression and secretion. The TCDA and TCDB genes are transcriptionally activated by TCDR. TCDR is influenced by environmental changes, such as changes in temperature and nutrient availability. It's important to get a sample when the patient has an infectious activity to have less possibilities of false negative results, and because of having a lot of microorganisms to get more DNA.
The mechanism of the test operation consists in the DNA extraction. When dwelling from the athletes and directly from the stool using QAA-AMP DNA stool minikit, DNA binds specifically to the silica hell membrane while contaminants pass through. PCR inhibitors are separated from the DNA by the Nivex buffer. The DNA extract was measured using nanodrop spectrophotometry at 2060 nanometers. Then we have the multiplex PCR assay. This is the assay for detection of TCDA, TCDB, TPI, triose isomerase using specific primers. The test was done by the epidermal cycler as follows: 1 cycle initial internalization at 94 degrees for 3 minutes, 35 cycles of internalization for 1 minute at 95 degrees, annealing for 3 seconds at 55 degrees, an extension at 72 degrees for 1 minute, and a final extension of 72 degrees for 5 minutes. The C. difficile strain ATCC9685 was used as a positive control for the 3 genes. The PCR produced electrophoresis is under condition of 1.5% agarose hill with 0.5 micrograms of milliliter safe strain, with 80 volts for 40 minutes, and the bands were visualized under UV light. LAMP assay was using 3 primers for the TCDB gene, 1 forward inner primer and 1 backward inner primer, 2 outer primers with 2 loop primers, look forward and look backward. It was performed with a total of 25 microliters. This mix was placed into a hot block plate at 60 degrees for 1 hour. They incubate at 80 degrees for 10 minutes to terminate the BST DNA polymerase activity. The ADCC 9685 strain was used as a positive control. This assay finishes with the detection of the LAMP products, with 1 microliter of SYBR green 1 0.1% observed on the UV light illuminator, showing the positive tubes in green and the negative tubes in orange. To confirm the results, an electrophoresis was performed on 2% agarose hill with DNA safe strain for 60 minutes. So let's talk about the principle of the test. Both MPCR and LAMP tests are based on nucleic acid amplification. LAMP, which depends on the autosomal strain displayed in DNA synthesis at an isothermal condition using Bacillus stereothermophilus BST DNA polymerase primary sets and a reaction buffer containing magnesium ions. 4 to 6 primers, forward inner primer and outer primer, backward inner primer and outer primer, were used to form the loops of DNA replication. The inner primers initiate LAMP by hybridizing the part of the elongate sequence to make a loop. The outer primers release single-stranded DNA due to the strand displacement DNA synthesis. This releases These DNA serve as templates for the next item primers producing a stem loop DNA structure. Multiplex PCR or MPCR is based on conventional PCR. It's used to denature and renature short segments of DNA using DNA polymerase I enzyme. It needs multiple set-specific primers that are utilized to detect several genes at once. PCR requires three main steps: denaturalization, annealing, and amplification. In the denaturalization phase, DNA is heated to dissociate the hydrogen bonds between complementary base pairs. The annealing process occurs cooling the denatured DNA at its temperature, allowing the hydrogen bonds to reform. The specific temperature is determined based on the physical and chemical properties of the specific primers used in the solution. Finally, in the annihilation process for DNA polymerase diffusion, it is essential to initiate replication. This process is repeated several times using a thermal cycler. This process results in the amplification of multiple copies of the target.
Stool samples were treated with alcohol to enhance the isolation of the bacteria. Then the specimens were vortexed and incubated at room temperature for 1 hour. The suspended stool is cultured in a selective medium and then incubated in an anaerobic jar. Finally, the isolates were identified by morphology of the colony and gram-positive appearance.
We can separate the MPCR test in 4 steps. The first one is the denaturation, where the temperature is increased up to 98 Celsius degrees to separate the DNA strand. Second is the annealing step, where the temperature is decreased from 48 to 72 Celsius degrees to allow the primers to base pair to complementary DNA template. The third step is extension, where the polymerase extends primer to form new DNA strands. Finally is the exponential amplification, where the process is repeated and the region of interest is amplified. Ranges used in this test are a buffer for the TOAQ enzyme, which is composed of three solution, hydrochloric acid and potassium chloride. Also, the oxynucleotide phosphide, which work as a building blocks of the DNA and are essential for the synthesis of new DNA strands. The forward initiator is a primer that binds to the negative strand of the DNA that's being amplified. Meanwhile, the reverse initiator is a primer that binds to the positive strand of the DNA that's being amplified. The DNA template is the DNA that contains the specific sequence we want to amplify. The DAQ DNA polymerase is an enzyme capable of replication DNA at high temperatures, allowing the amplification of specific DNA fragments in the PCR. For the LAMP test, all the process is done isothermic at a temperature from 60 to 65 Celsius degrees. The first step is a non-cyclic amplification, where the primer binds to the target DNA, generating a dumbbell-shaped stem loop at both ends, which is called the mother sequence. The second step is the cyclic amplification, where both of the ends of the strand begin to hybridize, forming structures of different sizes with different repetitions of the target sequence. Finally, there's the amplification of the DNA, where the loops provide additional starting sites for the DNA synthesis to accelerate amplification. These reduce their reaction time to less than 30 minutes. The reagents used in this test are kind of similar with the MPCR, using the DNTP, the buffer, and the DNA template. The change here is the betaine, which reduces the formation of secondary structures in guanine and cytosine-rich templates, and also sees the BST polymerase in strand separation. The magnesium sulfate reacts with the pyrophosphate that is formed as a product of the amplification, forming a precipitate that allows the test result to be observed. The BST DNA polymerase large fragment is an enzyme with the ability to displace an upstream DNA strand during the synthesis. Meanwhile, the primers used in this test are two external primers, F3 and B3, and two internal primers, the forward inner primer and the backward inner primer, and they work binding with the target sequence to help with the formation of the loops. The advantages for the PCR test are an early diagnostic confirmation, a simultaneous amplification and a detection during exponential amplification, and a high analytic attractor. For the LAMP, it does not require a thermocycler, works in real time, it is quantitative, it's highly sensitive and specific, and have a visual monitoring of the results. For the disadvantage, the MPCR test is qualitative, it is less sensitive, and it takes more time than the LAMP test. It also requires a thermocycler. For the LAMP test, it has a complicated primer design. It uses long HPLC grade primers, and the use of the test is restricted by regions and equipment availability in some countries. Both of the tests are confirmatory tests. In Mexico, there are some laboratories that can do the PCR test for the diagnosis of Clostridioides difficile, with a cost that varies from 1,900 pesos to 4,600 pesos.
That's all for our part, and thank you for watching.